Okay, hello. I decided that I would, maybe I should make this a bit bigger. I decided that I would just do a quick recording because a few people asked for uh, more information on the PCR worksheet. Most people did very well. In fact, this class did way better than most years. But I thought I would just go over it. And rather than just post a key, I think it can be more useful just to get the explanation along with it. My preference is to individually discuss with each group their answers and help clarify. But like in terms of time efficiency, that's not going to work. So basically, there were several aims for this worksheet. The first is for you to really understand the process. What is the primer? What does the primer do? How do you get the exact correct size, and all that sort of thing. But also paying attention. So paying attention to detail is very important. And one of the details of this one that I did try to signal by bolding and underlining is that you're starting not with one piece of double-stranded DNA, but two. So I didn't keep, giving only partial credit every time the mistake was made, but I did give some credit to the students who paid attention to that and included that in their answer. First question is just making sure you understand the convention that the 3' ends are the arrowheads and the 5' ends are the flat part of the arrow. Just, that is how we do it. I don't think any group got that incorrect. And then for the first 2 questions, these were already done for you in the lecture, but I am going to skim down and show them. So if you had 2 kb piece, I'm just going to show one for this case. And then you extended the forward primer binds to the lower strand, and the polymerase just goes until it gets to the end of the molecule. So that's going to be 1800. And then the reverse primer binds here. It's always going to bind there because of the sequence. And it is going to just, the polymerase will just go until it reaches the end of the molecule. So that is not well drawn. 1700. So what you're going to have at the end of that first one is you're still going to have your template, but you're going to have one each of the 1.8 and the 1.7 kb times 2, right? So you'll have 2 strands of 2 kb, one of 1.7, one of 1.8 for each double-stranded DNA molecule, and then so we're going to multiply that by 2. There's no need to draw it twice. Students kept asking me, should I draw it twice? And it's like, you don't learn anything from drawing it twice, but what you do learn is by the first drawing, and then there's nothing wrong with just multiplying by 2. It's interesting the kinds of questions I get, because they kind of make me feel like people think I'm like completely unreasonable and ridiculous, and hopefully you will not have that impression by the end of the semester. Although if you do, I guess I can't really control that. Next one. So basically, you've got 4 pieces of DNA in the tube, and then for the next round, each one of those pieces is a kind of template. So something can be made from each one. And it's just this part down here that we're looking at now. So we will make another 1800 from the original template, and we'll make another 1700 from the original template. But the 1700 that was already in the tube, the forward primer can bind to that and make a 1500 piece. And in this case, it stops at the end of the reverse primer because that's all the DNA it has to act as a template. And this is the first of our desired product. This is what we're trying to amplify. And then the pre-existing, the 1800 that was already in the tube, the reverse primer can bind to that, and it will produce the other 1500, and that's shown here. So what happens is, for each cycle we get one more 1.7, and we get one more 1.8 times 2, right? So that means now we'll have two 1.8 and 1.7. But we also have now 2 of that desired template. Okay, so we get to the next one, and now this is quite messy. I realized that after I had drawn it. But hopefully it'll be clear. I changed the colors to try to help. So the reverse primer will bind to the 1700 to make a 1500. It will also bind to the original template to make another 1700, which is it shown here or is it not shown here? The pale blue, yeah. So the pale blue is the extra 1700, and then so we get one more 1.7 here, and we get an additional. That's the orange 1800. No, it's the green. Excuse me. See, that's what happens when you make a messy diagram. So one more 1.8, and then we now have two 1.8s that pre-existed in the tube. One here and one here, and both of those can be templates for a 1500. And so for each 1800, we're going to get a 1500. So that's plus two 1.5s. And each of the 1700 pieces, there's one here and there's another one up here somewhere, there's another 1500 that gets made. So plus two 1.5 kb. But then the two 1.5s we already had will each give us additional strands. So that means each one of those, so we're going to have two more 1.5s. And let's see, is that correct? Yeah, and so basically, so what happens is what most students do at this point is they just start doubling the 1.5s. But you had only 2 of them here, and you're going to end up with 8 of them over here. So I've definitely missed something. And so it's more than doubling, because for every one of the 1.8 and 1.7s that you already had in the tube, you're also going to get a 1.5. And so these are the numbers you should end up with. So if you did it without multiplying by 2, the numbers, I put them in brackets here and then I stopped, because you can easily divide by 2 yourself to get the numbers you should have had. Because when I looked at your graph and so on, I didn't say, oh, the numbers, you know, if you used just one DNA, then I just graded it on the basis of that. And if you had used 2, I graded it on the basis of that. So the template never changes. And some people on quiz 3 are saying that the template like runs out during a PCR reaction. Like nothing happens to the template. It just keeps getting melted and then re-annealed. So no, it doesn't run out. It stays the same. We get what's called linear amplification of these pieces. These are our linear products. They are the ones that are replicated directly from the template, the original DNA that you put in, which is only used to make the linear pieces, right? And they increase, in the case of the question, by 2 each time. So when we talk about linear amplification, we're talking about a set amount being added each time in a reaction. So it could be 4 each time, 7 each time, 20 each time, whatever it is. That's linear amplification. The exponential amplification is when it doubles or more than doubles, because it's increasing in a proportion. And as the population grows, that proportion gets bigger and bigger and bigger. So you get this out of control, crazy amplification. So we start with, you know, at the beginning of the procedure, the first 2 cycles, there isn't any of the desired product. Then in the second cycle, we start to get a little bit of it. And then almost immediately, it starts taking off really hugely compared to the rest of the pieces. Like by the time we're at the fifth cycle, you can really see what that amplification looks like. Okay, so those are the numbers you should have had, and hopefully, I know that it's a bit messy, but if you try drawing it yourself and color coding it, you will probably find it easier to understand. And then your graph, I'm not going to put everything. So for full marks, you needed a title and you needed to label the axes, and the axes needed to make sense. Like you couldn't have, 1, 2, and then 50. You know what I mean? Because this isn't a reasonable scale, right? So you should have had something where the divisions are equivalent all the way along, and then you would see for the template a flat line, for the linear pieces none, and then a little bit, and then actually that's not a good. Let's just take that line out because it's a little bit more angled than that. Best not to add the two together, but like for each piece, the line looks something like that, and that's the 1.7 or 1.8. That's how they amplify. But then when you look at the PCR product, of course, it stays at 0 for a bit, and then at one point it's the same as the others. And then it's almost immediately starts to take off, so that you've got this, the line is like that. You can really see, this is, sorry, it's my non-dominant hand, so it's a bit of a mess, but you can see the exponential amplification very clearly. And part of making this graph was so that you could see why when we try to observe something on a gel, there's a certain amount of pieces that has to be there before anything is visible. Because when you run out your PCRs, there are lots of other little bits of, DNA in there. Some of your original template is in there, in that tube. But you don't see it because there isn't enough for you to see it. Okay. Yeah. And so basically, after 30 cycles, if you think about these guys, there's going to be 60 pieces of each of these strands. There's still only going to be 4 strands of these, so I've got these numbers wrong. And there's going to be literally like probably a billion or more than a billion in this one. So there's a certain threshold. You need around 20 nanograms to view on a gel. And I have talked about this in class a couple of times. There's a few places where I talk about the fact that there may be lots of DNA in the tube, but you won't see it because it hasn't actually passed that threshold of visibility. So the commonest mistake here was people said you'll see the 1.5 because it's more than the others, and that isn't precise enough, because if you had 5 million pieces of the 1.5 and 1.7 had 1.5 million, this piece, there's still more, but you would see both of them because both of them are beyond that, the limitation of what we can see. They've both passed that threshold. So for answers like this, I am looking for really precise and really specific answers. Just like if I say, what if your annealing temperature is too low and you tell me something won't work? That's not an answer, right? What exactly will happen under those circumstances? That shows that you know the thing, but it's also an example of good, clear scientific communication, right? Okay, so hopefully that's sufficient. And then basically the key for this is that for the reverse primer, it's going to be binding to this upper strand. So it's going to be the reverse complement of it. And when I'm looking for a primer sequence, I like to look for something that's kind of eye-catching. So 4 T's in a row. So in the other direction, I'm looking for 4 A's in a row. That looks like them right there. 4 A's in a row. G, C, A, T. Yeah, so this is it right here. 4 A's in a row, then a T and an A. So there's my sequence. When you're first starting out, you're going to have to rewrite it in the correct orientation and so on. So you'd be looking for A, T, A, G, A, T, A, G, A, G, G, etc. Okay. And for this, the most important thing is just simply your piece of DNA that you amplify is going to start here and it's going to end there. So you're really just counting the rows and then figuring out what's left out. So not this and not that or any of that. Okay. I did tell you the number of nucleotides per line so that you weren't having to deal with that. Okay, but this is the key thing. I don't understand in some ways why when you talk about like having a primer and then we talk about how they work with the DNA being added to the 3' end nucleotide by nucleotide, a lot of students then don't include the primer in the product, but that's what the DNA is attached to, and so that's a really important point to keep in your mind. And if you understand the process, then that would not be a mistake you make, and so it's important to think about that. Same for this one. It's probably exactly the same. GAGC, so it's there, and this is in the same location, I believe. So the sequence is a bit longer because there are introns in the genomic DNA, which there were not in the cDNA because they're spliced out of the mRNA. And if we aligned the cDNA sequence against the genomic sequence, if that was our genomic and this was our cDNA, what we would find out is the exact place and the exact size of any introns that we had. A lot of students said things like regulatory information, and there's not really a different, if we're comparing just a genomic sequence and a cDNA, the main thing we're going to see is placement of introns and how big they are. Okay, and then for this final question, when Jim and I were designing the primers, at the time we did not have cDNA sequence available, and we had to actually really spend a long time digging that out. But the primer we designed actually was partially exon sequence and partially intron sequence. So when we tried to use cDNA to amplify, it would not have worked because this sequence spanned this intron-exon boundary. And so, of course, it has a target it can bind to in the genomic sequence, but only part of it would bind on the cDNA, and that wouldn't be sufficient for PCR amplification. Okay, I hope that is clear. Feel free to ask any questions if you still have any.